Let me introduce to Christian Perez Shibayama. He is also a QFB, a chemist from the Faculty of Chemistry of the National University of Mexico. And he did also a master's and a PhD on immunology. And then he moved to Switzerland with Professor Burkhard Ludewig, and he has achieved lots of fantastic results on his research, and has been published in very important journals. So thanks, Christian, and please introduce Professor Ludewig.
Good morning, everyone. As Constantino said, my name is Christian Perez Shibayama, and it's a great pleasure, a great honor to introduce such a distinguished speaker, Professor Burkhard Ludewig. Thank you very much. Professor Ludewig is the head of the Institute of Immunobiology in the Kantonsspital St. Gallen in Switzerland. He's also the head of the cardioimmunology research at the University Hospital in Zurich. He studied veterinary medicine in the University of Berlin. Right? And obtained his doctorate degree in 1995, but then he continued his postdoctoral training in different places, Robert Koch Institute, the Hebrew University of Jerusalem, and very importantly, in the University Hospital in Zurich at the Rolf Zinkernagel Lab, which is a Nobel Prize in medicine. And it was the time when he met actually Constantino at that moment and starting a long-lasting collaboration with Mexican immunology. So it's a great pleasure to have Burkhard here. He's a pioneer in not only in stromal cell immunology, but also about cardioimmunology, and he's recognized as a global leader in this field. And Burkhard, it's a pleasure to have you here. I'm very much looking forward to your presentation.
Yeah, thank you very much, Christian, for the very kind introduction. You still can call me Burkhard, right? I know it's very formal here. But Constantino and Christina also, thank you very much for the invitation, and it's great to see you all here. I will, as mentioned, talk about cardiovascular projects that we have been pursuing over the last years, and it's about T cells, as indicated in the title, and I would say the extended title, or beyond the subtext, is that myocardial inflammation, it's immunology and beyond. So what are we talking about? And thank you, Adam, for introducing some of the complementarity. I will focus a bit more on the translational aspects of a particular disease. And since the audience is full of young researchers, young people, I think you might be in particular interested, because I will talk about myocarditis, which is worldwide, mainly Europe or in the Northern Hemisphere, a disease of young adults. So this is a case that a young lady who is in our study that we're following the disease. She was recruited into the study, and we're following her. She had, before she was recruited in 22, a post-COVID associated perimyocarditis, so a global inflammation in the heart. Well, there was pericardial effusion, so that's why it is pericarditis. She then recovered. Probably did not think that it's something serious, as many cardiologists in these days still think, at least in Europe and Northern America. But after last year's very severe flu epidemic, she contracted the same disease symptoms again, chest pain, very similar to the previous episode. She was then admitted to the cantonal hospital in Lucerne in central Switzerland. The ejection fraction, so the pumping function of the heart, dropped dramatically. Normal is everything above 50 to 60% of the percentage of the fraction of the heart that is pumped out of your heart with every beat. So she received an implant, which is a little pump into the left heart that further helps the blood to be pushed out of the left heart, but she then went, that did not help that much, she went into cardiogenic shock, was then transferred to the University Hospital Zurich, received, in addition to the impeller, to the little pump, an extracorporeal membrane oxygenation, because heart function was so low, so the blood had to be oxygenized. Still, there was no clear diagnosis. So what they do then, the physicians, they just give a bolus of prednisone. 1 day later, the endomyocardial biopsy, so which is via the jugular vein, a little catheter introduced, and then some small tweezers, forceps are inserted, and a very small piece of the heart wall in the right heart is taken. That showed CD3 positive T cells and macrophages, so that were established the diagnosis. Currently, what is done in these severe, or even slightly less severe cases, cardiologists throw everything on and add everything. So anti-IL-1 therapy, anakinra, and even intravenous immunoglobulin. Sometimes a very old method. It's called a anti-thymocyte globulin, which is in essence a polyclonal serum generated in rabbits to deplete lymphocytes in humans, and it's still given, and that's roughly what is out there for this disease. And luckily, it helped here many times before. So you realize in this disease, intensive care is very important. Keep the patient alive, and then do something that tunes down, right, that follows the theme that we had before, something that tunes down overshooting immune reactivity, without knowing how it exactly works. And that's, in essence, our mission. I'll summarize very briefly how we see myocarditis. In these days, we are more confident that this is true, and it's a simplification, of course, but immune checkpoint inhibitor treatment associated myocardial inflammation just shows us it's the T cells. So, and how do T cells, and I have been informed it's a mixed audience, so we are in between cardiology and immunology, so sometimes it's a bit simple, sometimes it might be a bit too complicated, so forgive me for that. So the simple view for the pathogenesis of the disease is that it's either, and we heard that before, it's the same theme, either it's the brakes off, for example, immune checkpoint inhibitors, then T cells directed against the tissue are switched on, so they are activated. It's CD4 T cells and CD8 T cells, or there is an accelerator function. This can be either molecular mimicry, because bacteria present similar structures that activate cross-reactive T cells. It could be any viruses. It does not need to be a virus that replicates or damages cardiomyocytes or replicates even in the heart. It can be any global immune activation, and of course it has been discussed as part of immune activation, and it's shown that part of the immune activation associated with vaccines, with any vaccines, can push the accelerator on the T cell activity, and that leads to the disease. How is it working in general? Again, for those who are not familiar with immunology, that might be a bit too much. For immunologists, it might be too simple, but I'll try to go through that here. So there is, for T cells in particular, they have to go to school. The school for the T cells is the thymus. When they leave the thymus and they pass all the exams there, they're called naive T cells. They get the further education and the instruction. Right, you use the military equivalence, right? So the army has to be instructed, and the instruction of the army and the expansion of the army, the basic training and expansion can be our lymph nodes. Then the T cells are called effector cells once they have received instructions, and they go to the target tissue. And here for myocarditis, so inflammation of the heart into the heart. So how does the education in the thymus work? What is important is that antigens, so structures that are recognized by the T cells, are either present or not present in the thymus. If they are not present in the thymus, so such as the immunodominant antigen in this disease, the myosin heavy chain 6, or the heavy chain, alpha heavy chain of the myosin. It's not in the thymus, and hence one part of the test in the thymus is, do you see cells? And if the answer is no, then these T cells can leave, and this is what's happening in the thymus for that particular antigen. That's why it is an immunodominant antigen, because it is not presented in the thymus. So there's the possibility that these cells are activated in the lymph nodes by, for example, dendritic cells or other professional antigen-presenting cells. If these T cells see and are instructed by this to react against this antigen, the myosin heavy chain 6, again here, for example, for the CD4 T cells, and then there are activation pathway circuits that lead to the expansion of these cells, and then they can go into the heart. How do T cells damage the target structure? So the heart here, it's either the CD4 T cells. They are not killers. They do not directly kill. They have their hitmen, and the hitmen of the T cells are the inflammatory macrophages that are activated, for example, by interferon gamma and other factors, and then they secrete factors that can damage the target tissue. Of course, not only eliminate bacteria, in particular intracellular bacteria, but it's a very similar mechanism. And CD8 T cells, they directly kill the target cell. So again, in myocarditis, it's again same theme as if we had talked about that before, but that's how immunology works. It's either to regulate, right, to regulate in a sense, to eliminate a damage or a pathogen, a threat, a perturbation, and at the same time to keep tissue integrity and to maintain tissue integrity. And this is disturbed if, for example, there's molecular mimicry, cross-reactivity. There is this balance is disturbed if there's excessive immune activation once T cells or antibodies are produced in a misdirected or produced in an excessive fashion, or what has been mentioned before, the equivalent is we've heard about that IL-10 is if IL-10 is not present, then there is a lack of regulation and then there's excessive tissue damage. So that's that's a lot to take in for the cardiologists. I don't know whether cardiologists are here. So when I present that at cardiology meetings, this is wow, right? This is a lot. For immunologists, this is of course our paradigm. But important, as Adam has mentioned, it is important for us. It is very important. Has been important to follow and to assess whether and to what extent these principles are really operational in the patient. So myocarditis in the patient, there are different, and it's an interesting disease. Why it's an interesting disease? It's a disease of young individuals. So the patients in our cohort see a hospital many times, first time in their life from the inside. So they have been hit first time. Second, it's interesting because there's quite a variability in the disease. So young individuals never seen anything before, and there's a variability from milder course and to a fulminant, the fulminant case I've highlighted. If in these days, because intensive care is so efficient and immunosuppression is present and there's more heightened awareness for the disease, if immunosuppression and maintenance of cardiac function is guaranteed, then there's a post-episode appearance. There's either resolution, there is recurrent, as in the young lady we've had, or a still more theoretical scenario that is chronic. It's just persisting. Or right in infection immunity, we know that as a persistent antigen, persistent pathogen-driven chronicity. How to study that, and I would also like to mention Christina, because it was Christina and myself 5 years ago who went to Zurich and set up the study, which is now supervised and as the clinical principal investigator led by Dörte Schmid, and 3 medical PhD students who are working with us, recruiting patients with acute and chronic myocarditis. The clinical definition has been, I spare you that, but the confirmation that it is indeed Myocarditis is done by either cardiac magnetic resonance, so CMR, and/or endomyocardial biopsies. And we focus today only on the variation on the disease presentation at presentation, so at day 0. We follow those patients for 3 and 12 months. So the recruitment, it's a rare disease. It's also, in terms of medical terms, it's an orphan disease. So there is no dedicated and cause-specific therapy. Overall, we have recruited until last month more than 100 patients. Overall, I'll summarize here. At baseline from Zurich and from St. Gallen, overall it's 79 patients, and from the acute myocarditis, 56. We even have already a 3-month follow-up. I'm presenting here, and it's always an intermediate summary. So in our study in Burkhard, I present data from 65 patients. I've mentioned that before, so the median age is 28, between 17 and 70. We are we're recruiting. After we know the demographics of our indicator of our patient cohort, we recruit selectively healthy controls. So same median age, same spread. As mentioned before, the majority of the patients is male, as in the controls. All of them are diagnosed positive by cardiac MR, and the left ventricular ejection fraction, or EF, I would call that in the following, is also varying, median 53, but some of the patients have a very low function of the heart. In clinical research, what do you need in addition? Of course, you have your main cohort. You always need a validation cohort. We're lucky to collaborate with the University Hospital in Tübingen, and they sent material, and we could explore the patient data from another 22 patients, so that the overall myocarditis cohort, the data I'll present, is 87. Healthy controls is still the same. What you can appreciate here is that the fraction of females is increased, and what is also changing and introduced the change in the cohort is because the patients in Tübingen showed a much more severe phenotype. They were all from the ICU, but all of that I spare you more details. That confirms in the validation cohort confirms what we conclude. Summarizing here the data, the clinical data from the aggregated cohorts from the 87 patients. Always in black the healthy individuals, and what you can see here is this is C-reactive protein, so the reaction of the liver to a disease in terms of inflammation. So there's a significant increase. Damage in the heart indicated by troponin I can be detected in almost all patients. NT-proBNP, or only B-natriuretic peptide, is a very important regulator that is immediately released and produced by the heart and by the kidney in turn at a situation of cardiac damage and volume changes, so requiring adaptation of the heart function. And as indicated before, one measure for the heart function is the left ventricular ejection fraction. 50, everything above 50 is still normal. Between 40 and 50, that's considered slightly reduced, already pathogenic, and everything below 40 is a severe impairment. So how do these parameters correlate? What is the overall disease severity that we see? NT-proBNP, so the natriuretic peptide, is a very good indicator for disease severity, as indicated by the very strong negative correlation with the left ventricular ejection fraction. As you can see here, the inflammation measured as C-reactive protein is not that well correlating. A quality criterion for the management of the patient is that the more severe the disease, measured as peak NT-proBNP levels, and the stay in the hospital correlates also perfectly well. So our patients are very well taken care of. So what is the reflection of the inflammation if C-reactive protein is not such a good correlate? We performed a multiplex cytokine and chemokine assay called Luminex and measured other factors by Eliza and Christina did all these measurements. So what you can see here, this is a heat map with the healthy controls in gray and myocarditis patient from the two different cohorts in red. The heat map shows immediately for the many different factors that you can measure in serum that there is a tendency to not have inflammation if you're healthy, but also in some of the patients, and some of the patients show different varying degrees of inflammatory markers in the serum. So what is the dominant phenotype that we see? There are a number of factors, T cell activation associated factors such as interleukin-2 receptor, or other T cell attractants such as CXCL10 and CXCL9 that are consistently elevated in the serum in the myocarditis, so the Zurich and Gallen cohort, and in the control cohort from Tübingen. Other factors such as hepatocyte growth factor, which might be interesting for you if you haven't heard about that, or ADAM. This is a factor that is produced by fibroblasts in the liver, but also the dominant source for hepatocyte growth factor are fibroblasts in the heart, and that shows a very strong relation and correlation with the disease severity. If we further delineate and segregate the different factors, the following pattern emerges. Namely, well, myocarditis is a T cell driven disease, so all T cell factors are significant. Almost all T cell factors are significantly elevated in the myocarditis patient. Of course, some myocarditis patient with a mild disease course do not show such strong systemic inflammatory pattern. And some of the patients show both a macrophage activation signature and neutrophil CXCL8, IL-6, and the macrophage chemoattractant. But still, these patients in their serum show the presence of T cell activation factors. So how to now mechanistically dissect this? It's a T cell driven disease. Of course, there is a certain degree of analytical proceedings that you can follow in humans. So what you need is a mouse model or any in vivo or in vitro model that reflects the disease, the pathogenesis. And this is what we even had established before. Namely, we cloned a T cell receptor specific for the myosin heavy chain 6. These mice, we call them TCRM mice, develop a spontaneous autoimmune myocarditis that starts early on, right after weaning. Approximately 50% of these mice progress to what is now commonly defined as inflammatory cardiomyopathy, if not. And we have to euthanize them if the disease is very, very strong at the age of 8 to 12 weeks, but only a fraction, which is not that dissimilar as what we see in humans. Of course, the humans we all rescue because they are in the ICU as if the ICU is available, as I've indicated. So these are recent data, recent summary from Anna, also Anna, who is working under supervision of Christina. Of course, control mice, they live, and the TCRM mice, so the myocarditis mice, we have to euthanize them at different time points post-birth. There are two main disease causes, either mild or severe. Mild, low inflammation, low fibrosis. Severe, high degree of inflammation, high fibrosis. So the red staining is collagen networks. In the mild disease, the heart looks almost normal, but there is always inflammation. And in the severe disease, there is dilatation and there is severe impairment. We also see this dichotomy in the phenotype in terms of fibrosis, if that is quantified. So the severe disease is this group, and the mild disease is this group. What you see here. So how is that reflected in terms of functional parameters. I've highlighted the left ventricular, the distribution of the left ventricular ejection fraction, so the heart function, the pumping of the function at admission at the first or second day in the hospital, and this is the spread of the ejection fraction in the TCRM animals. And the work has been done by Anna and Nadine. So this is an echocardiography of a normal, healthy control, and in the age-matched TCRM animals, what you can see there's an irregular beating. The ventricle, if you haven't seen these, that's what you see. This is the ventricle. The wall is thinned, and this is clearly a cardiomyopathy. That's a very busy slide. I will guide you very quickly through that. What you see, this is the time-resolved analysis of the ejection fraction, left ventricular ejection fraction in the TCRM and in the control mice. What happens early after weaning when all mice are still alive? So 24 of 24 mice are still alive. What happens is that there's swelling of the heart muscle, and that leads to changes in the function and with an significantly increase in the ejection fraction. So edema is known to affect ejection fraction, namely to increase also in humans. So after 6 weeks, I've indicated that some mice progress to the severe disease. So here it's 2 of the animals show a very low ejection fraction. We have to euthanize them. So hence we only have at that time point 22 mice left. Then we have an attrition in the cohort because more and more mice progress towards the disease, as you see here, and in the end there is a stable phase, what we call chronic myocarditis, where the values in the TCRM animals are not that dissimilar from the matched control animals. But what changes if we go further into detail? There is a measure, an echocardiographic measure for the cardiologist here that's simple. It's called strain, and that reflects the overall contractibility of the heart muscles of the heart muscle cells in different layers. I will not go too much into detail, but it can be longitudinal. So how does the heart compress in that way? How does the heart compress circumferential? And there's even a radial strain and a circumferential strain. What is the best predictor in these echocardiographic measures for the bad outcome? I spill the beans here. So it's the global circumferential strain, namely how does the overall ability of the heart to contract is a very good predictor at the age of 6 weeks for the progression to the cardiomyopathy, to the severe phenotype. I'll spare you some more of the data for the sake of time, also to have more time to discuss the conceptual summaries. So in acute myocarditis, so once the first wave of immune cells get into the tissue, there's an increase in the ejection fraction, the edema, and there is the myocardial strain is altered already early on. The overall performance in terms of ejection fraction and strain normalize if there is adaptation to the inflammatory response. So if there is low inflammation and low fibrosis. So there's an acute phase, and then there's a normalization in terms of functional performance of function in the heart. If there's a progression to the inflammatory cardiomyopathy, ejection fraction drops because there's more inflammation and more fibrosis, as we've indicated, and the myocardial strain, in particular the global and the circumferential strain, change profoundly. So what is the mechanism and how does it work? And here comes Christina. And all that has been done before, and of course with Christian together. And that's the paper in 2019 that we had in Science, namely clarifying how does the changes in the heart that we have explained now, namely that there's loss of cardiomyocyte, replacement of the cardiomyocyte by fibroblasts. Right now you've seen it, including the functional consequences. How is that caused? And I've mentioned it before, we spilled the beans. It's the T cells working with the macrophages. So T cells have to get into the myocardium to exert their function, to lead to cardiomyocyte death, right? Highlighted here. And they have to get into the heart. They have to be activated, right? We go back in time or in the process to the root, and one of the root process in the activation of the heart-specific T cells is their activation in the heart-draining lymph node. This is where the myosin heavy chain 6 drains into from the heart, and dendritic cells present the myosin heavy chain 6, so that the predominant pattern of activation is what is called, immunologists call the Th1 phenotype, namely interferon gamma is one of the signature cytokines. So there's imprinting of a Th1 phenotype in the heart-draining lymph nodes, and a dominant pathogenic factor in these tissue-damaging reactions is the ability to secrete interleukin-17. Where does that happen? And that's the major conceptual progress that was delivered by this paper, namely that they're cross-reactive. Bacterial peptide mimic peptides in the commensal microbiome, Bacteroides theta iota omicron. There's a peptide. You will see the structure of the peptide in a second. That leads to then imprinting in the gut environment of the heart-specific T cells, and we know they're heart-specific because we cloned the T cells following immunization with the heart peptide. So it's the imprinting of a Th17 imprinting that happens via bacterial mimicry in the gut. So I promised the structures. So this is the murine and the human myosin heavy chain 6. They are functionally identical, and these are the two of the identified mimic peptides. They're also very similar. This is the predicted binding to the IA of D. This is the class 2 molecule of the BALB/c mice, and this is, for example, the predicted binding of all these peptides to one of the more frequent HLA alleles in Europe, the DQ8. So overall, what do we conclude? It's the TCR repertoire, so the T cells that are present. Of course, the T cells need to be able to recognize their antigen. Microbiota have to be present to secure microbial mimicry, so antigenic mimicry, but of course microbes are always good to steer up the immune system in general, and as said before, the genetic background has to fit, because if the peptide does not, this is immunology, if the peptide does not fit into the groove, then the T cells have nothing to recognize. So it's all consistent as well. Now, swinging back to the immune checkpoint inhibition, Javid Mosley and his team, they were among the first to describe immune checkpoint inhibitor myocarditis, and what they found in the few patients that they had analyzed was that the HLA-DQB10301 was one of the major, they wrote, it's a New England journal, so everything's a bit focused. This is the major similarity between these two patients that they found, and this is one of the alleles of the alleles that is predicted to bind the myosin peptide and the mimic. Of course, that's all theory. How is that happening, and how does that all manifest in humans? So, due to we switch now back to the human, to the mechanisms. Is that really happening in the humans? So, for example, here this is published in Christian's Nature Cardiovascular Research paper. It's amazing that in these little snippets from the heart, we can detect 20, 30, 40% of the cells using single nucleus RNA sequencing being T cells. So that's hell of an inflammation in there. So if there is a high T cell infiltration, so a lot of activity, of course, a lot of T cells in comparison to other of these myocardial snippets with low T cell infiltration, the T cells are more activated, and of course, you would have guessed that Whenever there are activated T cells, the best buddies, the macrophages, the inflammatory macrophages are activated as well. And in these regions, cardiomyocytes are really stressed, and there are a lot of molecules known to indicate cardiomyocyte stress and death are upregulated in these areas. Of course, these are high-dimensional data, and I just summarize it, how we progress to analytically and hopefully therapeutically address this very interesting and we still think fascinating disease. So of course, we're cutting it, right? I'm cutting many edges now, and I'm sorry for this complicated display, but I'll guide you through. So this is a multiple correlation analysis. What you would agree and what we've seen is that there are very high correlation, significant correlation between the influx of inflammatory macrophages and T cells. There are a number of factors that are then upregulated, downregulated, co-regulated. And here we picked and highlight the correlation between inflammatory macrophages and T cell infiltration with the downregulation of the bone morphogenic protein 4. Significantly, this downregulation is significant in these data sets. In the paper, you will see the summary soon of the Nature Cardiovascular Research paper. We had a few patient data available to confirm that this is as well seen in the serum of the patients. So healthy volunteers have roughly 100 picogram per milliliter BMP4, bone morphogenic protein 4 in the serum, and it's significantly decreased in the myocarditis patient. Of course, now, right, you have these findings, and you're always eager to see whether after 2 years it's still true, and if you double or triple the number of patients and it's still true, so BMP4 is significantly lower. So this is our extending cohort with the matched healthy patients, and the regulators, because bone morphogenic proteins are molecules from the TGF-beta superfamily. These factors in the tissues have to be regulated in a very tight fashion. And this is achieved by, of course, on the transcriptional level, but also by the local secretion of inhibitors. And the inhibitors of the bone morphogenic proteins 2 and 4 are called, funny enough, gremlins, and there are 2 of them, gremlin 1 and gremlin 2, and fitting to what we had predicted and what we had seen was that there is gremlin 1 and gremlin 2 significantly increased in the serum of the patient. And if we even now calculate gremlin 2 and BMP4 ratio, it's a very good predictive value here in the ROC curve as an area under the curve of almost 0.9, which is very good. And if you just have enough patients, then it's the p-values, of course, exponentially good. So we can also visualize that, and thanks, Christina, for visualizing that in such a nice way. So the gremlin 2, and we've mentioned that the natriuretic peptide, there's a very strong correlation between the gremlin 2 presence in the serum and this very good indicator for the disease severity in acute myocarditis. And the healthy individuals, as you can see here with the BMP4 in the bubble plot, are all low for BMP and gremlin 2. And for human data, that's a very nice correlation, and of course, highly significant. However, this is again, we're swinging now back. Correlation is not causation. It's good that there's strong correlations, but it's not causation. So what did we do, and what did Chris and Christina do in the lab? They generated an antibody against gremlin 1 and gremlin 2, and it's the antibody that we continue with is called 14D102. And how does that work? So I've mentioned it. So bone morphogenic proteins and the gremlins are important factors in the tissue. The receptors are called bone morphogenic protein receptors, BMP receptors, and if everything is running fine, and what is the function of many of these TGF-beta family cytokines is just signal the tissue everything's good, life is good, right? Have a tea, have a coffee, and just do your work and do what you're paid for. So that's homeostasis. Under inflammatory conditions, for example, in myocarditis, all kind of causes we will not go into, then the transcriptional activity of BMP4 and BMP2 is reduced, so there is less BMP4. Gremlins are even a bit elevated, so there is less BMP receptor signaling. That is, in immunology global terms, inflammation or danger that tells the tissue you are not in homeostatic conditions any longer. And at the locus of the fibroblast, that indicates and that fosters the activity of an inflammatory fibroblast. Well, now, if you intervene and you give an antibody, an inhibitor of the inhibitors, then you restore BMP4 availability, and with that, BMP4 receptor signaling. So I'll cut a long story short and show the key data by where Chris and Christina have treated TCRM mice, so the myocarditis mice with the antibody right at the onset, when the first wave of immune cells is attacking the heart, and you remember when there is edema, so acute myocarditis, and for 4 weeks, and after 4 weeks, the infiltration with immune cells is reduced in the heart, and what is important, the pathogenic cytokines, interferon gamma and IL-17, are reduced in the cardiopathogenic T cells. And what is important as well, the availability in the heart, so the heart tissues were minced and protein extracts were measured, and BMP4 concentration. There was only 3, right? Yes. I've got another 10 minutes, so that's very good. You know, I'm from Switzerland, although originally from Germany, I'm from Switzerland, and we are obsessed with time. If you've ever been to Switzerland, there are clocks everywhere. You just don't need to look. I don't have a watch. There are clocks everywhere. So whenever the clock rings, I get, am I too late? But and don't worry, ask Chris and Christina. If the bus is late, they really get nervous after only 15 years. I don't know how it was with Constantino, but you swung back, right? You're more relaxed now. So I'm still relaxed. So thank you very much for the nice intermission. So what happens now with the antibody? So we restore BMP4 availability, and with that, we'll not go too much into detail, fibroblast homeostasis. So we counteract the activity of an inflammatory fibroblast, which is conceptually still a bit new. People immunologists in particular, are of course familiar with the inflammatory macrophage, but that fibroblasts, the truly tissue-resident cells, can switch from one instant to the next into an inflammatory cell. This is what we are addressing here. So the next step, of course, is what do we do? And this is work of Hongwei Cheng, who's in the lab. If there is already a novel potential therapeutic principle, and you want to carry it into the clinics, and you have a mouse antibody, of course, you cannot give that to humans. The next step is to humanize the antibody. So we collaborate with a Swiss company called Lonza, and they have algorithms, techniques, and everything that is required for a process that is called CDR grafting, so the complementary determining region of those amino acid snippets from the antibody that determine the antigen binding have been transferred from a mouse antibody into a human antibody. That's the visualization of the antibody that has introduced a few more mutations. And what is always important to address and to assess whether a humanized antibody or a human antibody that has been extracted from the human repertoire is really functioning and doing what you want. Of course, it's helpful to have a mouse model that reflects and is very similar to the disease process that you would like to treat in humans. And the possibility that we have here is if we take mice that lack, so BALB/c, white mice, very similar in their genetics to the TCRM animals that lack T cells and B cells, we can transfer the cardiopathogenic T cells from the TCRM animals, so the cardiac myosin specific T cells. These T cells expand, they go into the heart, I've described that before, and they cause myocarditis. If we start the treatment with the antibody even before, and here it's head-to-head comparison of the murine antibody and the humanized antibody, and wait for 4 weeks. What you can see here in the isotypes, so in the control antibody-treated animals, there's inflammation. Treated with the murine antibodies, it's reduced. With the humanized antibodies as well. And what is important, that we get a significant reduction in the cardiopathogenic cytokines, namely interferon gamma and interleukin 17. So Christian and Christina, this is also, of course, right. The mission is to the reintroduction of you two guys into this microenvironment, into this ecosystem. So Christian and Christina have summarized that, and that was published last year in Nature Cardiovascular Research. And Pilar Martin from Madrid has written a very nice news and views on that. And to summarize that, overall, what I wanted to tell you, in the myocarditis and myocardial inflammation, it's the T cells, and it's the modification of the microenvironment, namely here at the locus of the fibroblast, that determines the extent of the disease, and at the same time offers opportunities for immunotherapeutical intervention. Namely that under homeostatic condition, BMP4, and probably other factors as well, produced by fibroblasts, signal into the environment, everything's good. Do your work, cardiomyocyte. Because why is that important in the heart? Of course, the work is done by the cardiomyocytes, but they are really hard workers. Many, many times per day is your heart beating and the cardiomyocytes are contracting. Plus, whatever you hear, cardiomyocytes do not regenerate. Once you lose them, you lost them. So the whole system has to be geared towards cardiomyocyte maintenance, survival, and well-being. That's simplified. So, and one of these well-being factors is bone morphogenic protein 4. Under conditions of inflammation, there's a reduction of that pathway. Fibroblasts are switching into inflammatory mode and foster inflammation whysoever. And the antibody that the principle that we have discovered is to inhibit the inhibitor, namely an anti-gremlin 1 gram2 antibody, and that rescues, that antagonizes the disease and leads back to homeostasis. So I leave it here. I thank the whole team in St. Gallen. We have not that high mountains, and there are no volcanoes, at least in Switzerland, but the mountains are still very nice, as you can see in the background. This is one of your pictures, right, Christina?
Christian took it.
Christian took it, right? So thank you very much, muchas. So this is on one of the lab trips. This is the core group that is working with me. Christian, Christina, Hongwei I've mentioned, Nadine and Anna I've mentioned, Emily, Isabella as well. And we could not do the work with the team in Zurich with the support of the head of the clinic, Frank Rožička and Dörte. And there are other cardiologists and colleagues from all over the world who support our research, including the European Research Council and the Swiss National Science Foundation. And all that's left now is to thank you for your attention. Thank you.
Antes de darle la palabra a Christian, quiero comentarles que ellos están muy orgullosos, son egresados de nuestra unidad. Ellos estudiaron ahí desde la licenciatura hasta el doctorado, y luego realizaron todos estos estudios, como lo mencioné. Y no solo ellos, sino más mexicanos, investigadores jóvenes, fueron a sus laboratorios a continuar su preparación. Entonces estamos muy, muy orgullosos y contentos, y ojalá los podamos reclutar en un futuro próximo al instituto. Thanks, Burkhard, for this great talk. Now I'm going to handle to Christian to coordinate the questions.
Thank you for your presentation. It was fantastic. I have 2 questions. The first question is, what do you think that the T cells can change your phenotype in different severity levels in myocarditis disease? And the second is, do you think that inflammatory fibroblasts in this disease can drive these infiltrate of inflammatory T cells?
Yes.
Thank
you. The answer is twice yes. I elaborate a bit more. So T cells are the drivers, right? Of course, there are different ways to affect T cell activity. However, Our approach is more to go further to the root of the process in the tissue, because meddling with T cells has advantages and disadvantages. And we firmly believe that the future approach is to modify the microenvironment at the locus of the fibroblast. So the double yes, well, T cells drive, but the supportive infrastructure determines to what extent and how the T cells are acting, and of course together with the macrophage.
Sorry, and the last question. Do you think that therapy over microenvironment like fibroblast is necessary for this type of disease?
Well, that is, thank you very much. The answer again is yes. Interfering with T cell activity overall or with immune activity has been very successful, right? This is still what's given to these patients, prednisone initially, right? So you just hammer it, right? To come with the analog, with the war analog, right? That is, in terms of immunosuppression, the prednisone is something between between a very big bunker buster and a small nuclear bomb. So that's very severe. Still helps, but is not a real long-term alternative, because inflammation in the heart in particular, right? Now we save these patients. The mortality was much higher. But the challenge is, if these, of course, identify the patients who are susceptible, in addition to the acute myocarditis, there's most likely, and there are very good indications, that inflammation in the heart muscle, is a major driver of more than acute myocarditis, of many other disease conditions. But right, as a very nerdy scientific approach, we prefer to study the processes in a patient cohort that is not at the end of a potentially immune-driven process, such as heart failure patients, cardiomyopathy patients, age 50, 60, 70 something, and with a very, very long list of medications already. So hundreds of confounding factors, almost impossible to delineate, to dissect pathogenic mechanisms. And hence, the focus on the acute myocarditis, where I've started, because these are young individuals, young patients who see the hospital first time in their life. And of course, cardiologists are asking, yeah, but we have to treat our heart failure patients, our cardiomyopathy patients. Yeah, of course. That's why the clinical studies in cardiology need 100,000 patients to see a minimal effect. And even in particular, the immune approaches many times have failed now. Retrospective analysis, sub-patient group analysis, oh yeah, but there are some patients that benefit. But the design has to change. Right? You can't run these clinical trials in cardiology in this way any longer. And it has to adapt to what is done now in rheumatology, or done in rheumatology for a long time, and what is done in oncology as well. Right? We have to treat and separate the patients according to disease mechanism, and of course to treat them early if there is a clear suspicion and clear indication that it's immune-driven. T-cell-driven, macrophage-driven, TNF-driven, or whatsoever. Sorry for the long answer, but that is from the bottom of my heart.
Hi, Burkhard. Thank you for that talk. I come from the group A strep field research, which then goes on, repeated infections can end up in rheumatic heart disease and very young patients, as you say. We don't have very clear, we know it's molecular mimicry. We haven't really identified the specific peptides that can be driving this progression of disease. But I'm curious about this IL-17 or Th17 imprinting. Do you know if there's something in particular in young individuals that can, have a more enhanced Th17 imprinting rather than adults? Because, yes, as you said, it's like a young people.
No, we don't have data But I would argue, no, there's in principle no difference.
Yep. Right.
What is different in the more adult or more aged population is that the T cells overall get more experience, right? The thymus stops to work, there's no more output, and there's this clear selection of antigen experienced T cells. So the overall degree of what we now would call regulation is increased, so there is more regulatory activity in the more age-advanced population. So there are differences. But in principle, they still, I can still produce IL-17 or Th17 cells, because otherwise the bacteria would have eaten me alive.
Yep. Thank you.
Right, Adam? That's how it should be, right? Th17 should still be there, even at the advanced age, until the system breaks down. But there are questions, sorry.
Thanks for your presentation. My question is, there participation of autoantibodies in this pathology?
Sorry, there's?
Autoantibodies.
Autoantibodies. Oh yeah, autoantibodies are always present. The question is, to what extent do autoantibodies contribute to pathology? Right, we've heard that before. Antibodies are able to mediate via Fc receptors, complement activation, platelet aggregation. That's all possible mechanisms also affecting the heart. In the myocardial inflammatory disease in particular, antibody-driven effects are probably very rare. First of all, because B cells are very rare in the heart, and at least in the studies that have been done in mechanistically, autoantibodies or antibodies directly seem to affect the heart in a very rarely. So answer is yes, but probably not as severe as in other conditions. But still that has to be explored further.
And the second is, you are evaluate the Tregs?
Yes, we are on that, yes. We are confident that they follow the general rules with age increasing, and that disbalance.
I mean, the function is normal or affected in the context of the pathology?
That's it's overall right. We prefer not to look at Tregs alone, because it's the balance between the T cells that drive the disease and those that try to moderate and regulate the disease. So we always look at Tregs. So overall, in the T serum animals, the Tregs are not absent. On the contrary, 30+% of the cells in the inflamed heart and even in the severely diseased hearts express FOXP3 and qualify for regulation. But their activity is overruled by the Th1 and Th17 cells. And we would assume this is very similar in the patient.
Thank you.
So, Professor, you discussed 3 major factors that contribute to high risk for cardiomyopathy, which is like the TCR, microbiota, and genetics. Is there like a certain factor that we can work in for the future that can contribute the greatest to a decrease in the progression of the disease? And for the treatment,
please put your microphone a bit up. So
should I repeat, or?
I think I got it.
Okay. So also, should we use the anti-GREM for the treatment, or should a more generalized immunosuppression be used? Because if our goal is to save the cardiomyocytes, we can use the immunosuppression to get a stronger response, even though there is a higher risk of getting more infections. But I think cardiomyocytes, more cardiomyocytes can be saved that way.
Exactly. So the way initially more broader, particularly in the very severe cases, the prednisone or maybe a bit more specific anti-inflammatory, global anti-inflammatory treatment is very important. Right? We have to cut it. This is right. Clinical experience has shown that works. But immune responses will continue. And there is a continuous, right, if you get it once, it's very likely that it's still cooking, right? It's smoldering. And we still have to find out, in particular in cardiac diseases, to what extent immune-driven smoldering inflammation is affecting the cardiomyocytes. And we see that it's a cut initially with immunosuppression, and then at the end find the most suitable therapy to maintain the environment most supportive for the cardiomyocyte. I think we would agree on that.
Thank you.
Great talk. And you show us the importance, the relevance on controlling this inflammation. I think this is great. But I want to go maybe a bit further on the resolution of the inflammation. Then I think there is something lacking there in the heart to resolve, and then it goes, instead to regenerate, it goes to the fibrosis. In our preclinical model of the axolotl, we find lots of, and this axolotl, the Ambystoma mexicanum, it regenerates the heart. And then we find lots of B cells there. I mean, well, not exactly the B cells, but we find the VH and the genes at the moment. And I was thinking if there will be some role, and maybe we are lacking of protectins, resolvins, and all these factors that help, after controlling inflammation, resolve the inflammatory process. And
so resolution of inflammation is very important. We heard that before also from Adam. So control of the degree of inflammation in the heart in particular. It's even then different from or the equivalent to the loss of neurons in the CNS is, so how does the CNS cope with the loss of neurons, because they cannot be replaced. So they grow more and additional connections, right? Axonal growth, additional connections. So how does that work? I would say compensation in the heart. The compensation is first that the fibrosis is not always bad. So the fibers that are generated by the fibroblasts that replace the cardiomyocyte are good because they secure the forced transduction, right? The cardiomyocytes have to contract, and if there are few of the guys are missing, someone has to bridge it, because otherwise there's no forced transmission. So that's what the cardiomyocytes do. Second, the fibroblasts and the fibrosis, in that sense, there has to be a remodeling in the heart so that maintaining the remaining cardiomyocytes that grow simply in size. What is the compensation? Right? In the CNS, it's more axons and more other connections, and in the heart, the remaining cardiomyocytes are just growing in size. So the infrastructure, right, coming back to what do we have to do? We have to optimize in long term the infrastructure in the heart to find out what keeps the cardiomyocyte happy, healthy, and protects the cardiomyocyte from further damage. That's also coming back to your question, and maybe answering partially what you just said. Overall, just starting from basic tissue biology and from the function. Good. Adam.
Hi, thanks. Really, really interesting talk. So my first question is the balance between the Th1 and 17 and in a systemic organ. And have you, I don't know, back-crossed the mouse onto like a TBET or something like that, or raw gamma, and then done adoptive transfer studies just to see the relative contribution?
Yeah, we started with interferon gamma, which is it's a very interesting cytokine, right? It's pathogenic at the beginning, but if the interferon gamma or the interferon gamma receptor is lacking, at later stages of T cells develop as cohorts. They acquire on a cohort level different properties, and they adapt. So at later cohort maturation, differentiation, interferon gamma becomes regulatory and holds back. So yes, we did, and that's what you do in research, right? All the young guys here. The more you find out, the more questions you get. So yes, the cytokines change, regulation is kicking in order to keep, and it's very similar to what you observe, in order to keep the balance between protection, and here in the tissue, protect the cardiomyocyte at all costs, and at the same time get a not minimize, but keep the inflammation at a reasonable level so that, again, the heart function is not affected.
And this is probably a naive question related in, I was actually quite surprised to see such a IL-17 contribution in a systemic site in terms of say to pathogens, to Salmonella, you'll virtually never see any meaningful systemic IL-17 or Th17 response. Mucosally, you will.
Yeah. So one explanation that we delivered for this question, surprise initially, right? IL-17 is important, is the cross-reactivity and the additional priming site, and it really happens that the heart-specific T cells proliferate in the gut-associated, in the colon-associated lymphoid tissues, right? So there is priming, and once they're primed, they also leave the colonic patches, and then once they are instructed, they can go into the heart. So that is one. And we have other mechanisms elaborated or on the way, how the Th17 and where the Th17 imprinting in this particular context is following induction is also maintained and even amplified.
And just finally, a very provocative statement. You know,
I love that.
So MS, rheumatoid, but both of those are very, very strong T cell mediated component. But you treat with rituximab B cell depletion. Mm-hmm. So does that mean, you know, and probably that works, possibly targeting through antibody, but probably through regulatory B cells, which go not quite hand in hand, but you know they know where each other live. And do you think that is the same, that the T cells in this not just are being pathological, but are being puppet masters? So if you target the the T cells instructing B cells to do other stuff, you know, generic. Mm-hmm. So if you knock them out, you can maybe regain balance, or
So warning, it's a longer answer, right? If you are, and this also relates to what we have with the regulatory T cells, right? If you assume that there is one dominant factor, and you call it regulation on the level of T cells, macrophages, B cells, whatever, and then you look for it, and then you find B cell regulation, and then you have your puppet master, right? But that's what we do. Right? We do the same. We postulate that there are inflammatory fibroblasts, and we find them, right? Because we had indication. The question is now: is that the only role? Most likely not, because B cells act on many different levels in the immune system. Without B cells, secondary lymphoid organs are not well organized, and you generate a relative immunodeficiency. Because the B cells are very important to maintain, for example, the splenic, the marginal zone in the spleen. Right? Without B cells, so you reduce the B cells, then the splenic structure is screwed up. Sorry, excuse me. And it's the same for the lymph node. So that's one. So it's the systemic immune reactivity. Second, B cells are very important antigen-presenting cells, and they're very important in their interaction with T helper cells. I would even say, well, they're at least as important as the famous dendritic cell. You may call them CDCs, PDCs, regulatory, whatever. Right? What is ever fashionable now, right? So the B cell therapy, and of course the experimental medicine that then shows that, yeah, well, if you cut, and coming back to the to global immune suppression, I think anti-B cell therapy is another means of very global immune suppression on many different levels. And it provides excellent opportunities for us as immunologists to postulate our regulatory cell population of the day, and to have a flourishing lab based on that. Sorry, that was sarcasm off. But that's the way it is, right? That's we do it as well, right? We work on inflammatory fibroblasts, and we focus on that, and we can because we need these conceptual narrowing, because we have to come up, right? Can we talk to the youngsters a bit, right? Because we have to come up with questions, we have to focus, and we have to ask clear questions. Because if we say everything is global, everything is, right, then we don't achieve. So in a very positive sense, yes, B-cell regulation exists. B-cell regulation is also affecting T-cell activity. But there's more to it. Sorry, I declared the long answer.
Thank you.
Oh, do I get a
Thanks.
prize? Thank you very much. Thank you. Thank you very much.
Thanks, Burkhard, for this great talk. Thank you, Adam. We really enjoyed your sessions and that you share with us this your part of your scientific journey and the latest developments of your research. I mean, I met these guys before the snow of time was fluttering their temples, and it's great. They have been studying many different subjects and the evolution of the research and all the impact that has. I want to thank for the impact you have made here in Mexico with my students, with the students of other Mexicans, and all promoting research and help us to put forward immunology in Mexico, and in particular to take all these advancements to improve the healthcare of our people here. So thanks a lot, and thank you all for your presence here. Thank you for to the people that follow us from YouTube, Facebook, and the Academy website. It's great to that you were here, and thanks a lot. Have a great weekend. Para los que están aquí, profesor Ludewig y el profesor Cunningham van a estar unos minutos por si quieren platicar, acercarse a hacer algunas preguntas. Vamos a estar unos minutos por aquí, después vamos a tener unas reuniones, entonces va a ser momento para que se puedan acercar. Gracias.